Welcome to you all today for the next journal club. I would like to introduce Dr. Arturo Roca-Rivada. He defended his PhD thesis in 2013 at the University of Santiago de Compostela in Spain, where he worked on novel adipokines and myokines implicated in physical exercise and obesity. Then he made a move for his postdoc to Paris to work at the Institute Cochin, supervised by Dr. Julie Dam, where he worked on deep characterization of two different knockout mice models, finding that the whole body deletion of a protein involved in leptin signaling protects from obesity-associated deleterious metabolic alterations. And he also worked on the identification of an canonical G protein coupled receptor. And then he made a second move for a postdoc in 2020 to Brussels, where he is now, I think, at the ULB Center of Diabetes Research, supervised by Dr. Isaac. And I think that's also the research where he will be talking about today. So please share your screen, Arturo, and the floor is yours.
Okay, thank you for this kind introduction. So, okay, can you all see my screen?
Yes, and we also see the pointer, yeah. Okay,
so, well, thank you so much for this, for inviting me to present our work. For me, this is really nice because we had the opportunity to defend our work and to have the feedback of the scientific community, and also because this is just the surface of work that is still ongoing. So this is just the beginning. So I'm kind of excited of presenting our findings. So this study entitled Inhibition of the Type 1 Diabetes Candidate Gene PTPN2 Aggravates TNF-α-induced human beta cell dysfunction and death, that it was recently published in Diabetologia. So this is the work that I'm gonna present today. So as a brief introduction, this is just a simplification of a more complex process. But when an external aggressor attacked the beta cells, for example, the cytokines, beta cells respond in different ways. For one side, the beta cells, they start to activate the apoptotic signaling that can lead to beta cell death. And on the other side, the beta cell is capable by the expression and secretion of chemokines and cytokines and the presentation of modified antigens to recruit immune cells and activate them. And these immune cells, what they do is to start also expressing and secreting cytokines like TNF-α or interferons that they also attack the beta cells, creating a vicious circle that leads to beta cell destruction. Of course, this doesn't happen in a healthy cell because the beta cell has a mechanism to modulate the activation of these pathways to counteract this cycle. One of these mechanisms is through a specific phosphatases that inhibits the pathways, for example, PTPN2. I will talk about that about this protein later. But first, I'm gonna focus in one of the aggressors. As you know, type 1 diabetes is diagnosed really early in age. And this suggests that maybe the autoimmune process can start early in beta cell development. So this confers more importance to the innate immune system and also the early response cytokines like the interferons or the tumor necrosis factor alpha in the progression of beta cell failure. According to this, in a recent study published in 2022, after the Ackenberg and his colleagues, they perform a classification tree analysis of 2 different cohorts of type 1 diabetes patients, and they managed to divide these cohorts into 7 subgroups with specific signatures. So from those subgroups, the groups that included the patients, the most youngest patients, and with more aggressive forms of type 1 diabetes, they show a clear signature of high levels of TNF-α in blood. But more important is that in 2020, it was published a phase 2 clinical study in which young patients with type 1 diabetes were treated with a TNF-α inhibitor called golimumab. And after this treatment, these patients show promising results regarding C-peptide preservation and also a better production of endogenous insulin. So this is about one of the beta cell aggressors. But what about the mechanism that beta cells has to avoid the cytotoxic effects of these aggressors? So one of the proteins involved in this mechanism is PTPN2. The interesting thing about this protein is that in recent years, several polymorphisms of this protein has been associated with the development of type 1 diabetes. And the interesting thing about this protein is that it's a specific tyrosine phosphatase that has multiple targets that are implicated in the control of beta cell physiology and survival. In our team, we study a lot this protein, and so we published several papers regarding the functions of PTPN2 in beta cells. And we discovered that PTPN2 is able to modulate variant, but also type 1 and type 2 interferon responses in beta cells, basically by the dephosphorylation of STAT1, deactivating the JAK-STAT pathway, and this protects the cells from an excessive cytotoxic effect of these cytokines in beta cells. So with all this data in mind, our hypothesis was to see if the defective variants of PTPN2 are able to sensitize beta cells to early response cytokines, including interferon alpha, but also TNF-α. So for that, the first thing that we do was by using specific siRNAs to silence PTPN2 in different human beta cell models. So we work with endoc-PTH1 cells, disperse human pancreatic islets from donors, and inducible pluripotent stem cells that we differentiated into islet-like cells. So after the silencing of PTPN2, what we do was to treat the cells 48 hours with a combination of cytokines, interferon alpha, TNF-α, or the combination of interferon alpha plus TNF-α at the same time. And then we measure beta cell death by staining the cells with host and propydion iodine staining. And when we did that, what we observed is that the cell silence for PTPN2 after the treatment with TNF-α, that induced an increase in beta cell apoptosis, but also when we treat the cells with interferon alpha in all the 3 models. And the most interesting part is that when we combine both cytokines, we did not observe a potentiation effect in beta cell death. And this was suggesting already that the pathways that the interferon alpha and TNF-α activates and leads to beta cell death may be common for both. So what we did next was to check by mRNA expression different markers of beta cell development, like PDX1 and insulin. And when we saw that in the 3 human beta cell models, we saw that in TNF-α drastically reduced the expression of PDX1 and insulin in all these models. and also interferon alpha and dog beta H1 cells, not in the other models. That is interesting. So this also indicates that the deleterious effects of TNF-alpha is already more intense in beta cells than interferon alpha. That's a curious thing that we saw here. But it is just mRNA expression. Wanted to go and check the functionality of the cell. So for that, what we did was to perform a glucose stimulated insulin secretion, in this case in end-dog beta H1 cells, and we observed that when we silence the cells for PTPN2, the cells still respond to glucose, but they have a lot of problems to produce insulin, and this could lead in the future to a beta cell malfunction. So we wanted to, after all these results, we wanted to know which specific pathway was involved in the effects of TNF-alpha. And we wanted to compare it with interferon alpha, as I said before, that we thought that they may be sharing a pathway in common. So as we studied in the past the effects of interferon alpha in a context of PTPN2 silencing in beta cells, we knew already that these effects were mediated through the beam expression. So what we did next was to do a double silencing of PTPN2 and beam, and then follow a similar strategy than before. Treat the cells with interferon alpha, TNF-alpha, or the combination of both for 48 hours, and then measure for beta cell death. And what we observe here is that when both proteins are silenced, the cells resist the cytotoxic effect of interferon or TNF-alpha in both models, and dog beta H1 cells and iPS-derived islet light cells. But going further into into this pathway, because beam is really upstream of the pathway, we wanted to go what PTPN2 was doing, and we knew already that for our studies with interferon alpha, that JNK1 has something to say there. So what we did next was to do similar experiment. In this case, we silenced for PTPN2 and GNK to see if also TNF-alpha deleterious effects were also mediated through GNK1. So after doing that, we observed that when we silence PTPN2 and GNK1, it could also inhibit the cytotoxic effect of interferon alpha and TNF-alpha in both human cell lines. So last thing that we did, and this we realized by going to the bibliography in studies performed in colorectal cells, in fact, because in these cells it was described that the PTPN2 or GNK1 could be a substrate for PTPN2. So we thought, okay, let's see if it also occurs in beta cells. So we perform a immunoprecipitation in end-dog beta H1 cells, and we observe that GNK1 immunoprecipitates with PTPN2, showing that there is a physical interaction between these two proteins. And then when we check by immunoblot the GNK pathway, we saw that the silencing of PTPN2 increased the phosphorylation of JNK1 when the cells are treated with TNF-alpha, and also the downstream of the downstream protein beam. It also increased the phosphorylation. But when we check for the upstream protein MKK7, upstream of JNK1, we did not see any differences. So what we conclude with this is that PTPN2 is directly affecting the phosphorylation of JNK1. That leads to the phosphorylation of beam, and this leads to beta cell death, and that this pathway is common for interferon alpha and TNF-alpha effects. So as a conclusion, in the different studies published really recently, TNF-alpha inhibition is able to delay the progression of type 1 diabetes, and also the circulating TNF-alpha is associated with aggressive forms of the disease. And here in this paper, we're showing that the candidate gene of type 1 diabetes, PTPN2, is a key regulator of the deleterious effects of TNF-alpha human beta cells. So it is conceivable that patients that carry risk-inducing PTPN2 polymorphisms may particularly benefit from therapies that targets TNF-alpha. And that's it. I would like to thank the people from my team that are fantastic, the team led by the Professor Dasil Sirik, and also the team of Miriam Knopp and the team of Piero Marchetti, that they help us a lot for this work. Thank you so much.
Great, thank you, Arturo, for this nice presentation and the clear explanation of your paper.
You said in the beginning that golimumab, I hope I pronounce it correctly, was used in early type 1 diabetes. Do you know what is happening there? Is it now used in kind of earlier stages of type 1 kind of risk, or is it still in a clinical trial, or do you know what is going on?
Yes, so there is a follow-up study that it was published, I think, last year or something like that, that they stopped giving this inhibitor to the patients, and these patients they still have beneficial effects than the patients with the placebo, even without receiving this compound. I don't know if they are continuing with the test. I don't know. What I can say about this compound is that this is already approved and is used in the routine for rheumatoid arthritis. So that's a great advantage of this kind of drugs.
In addition to the question Claudia asked, are there any systemically effects from this TNF treatment? Because you only want to treat locally, right? Or you would ideally only want to treat the beta cells, I guess?
That would be the idea, because the problem is that this kind of drugs, they are attacking the immune system. So they have some side effects related with infections. And so yeah, it's a bit tricky.
Yeah, so there's some optimization might be needed there, right, to deliver it locally.
Yeah.
So are there any questions from the audience? You can either speak up or put your question in the chat. I see Ben raised his hand.
Hi, Arturo. Thanks for a lovely presentation and a great paper. How do we know the specificity of this phosphatase? Is it directly targeting J&K only, or is it quite broad range? So, or could a phosphoproteomic screen, for example, be useful to tease out what it's targeting?
It's not too specific. It's only specific for tyrosine proteins, but it targets also STAT1, STAT3, JAK1, I think. There are several substrates of this protein, but most of them they are related with the similar pathways.
Yeah,
that's the interesting part.
So you'd expect not many off-target effects if you're inhibiting the same pathway, right?
Yeah.
Interesting. Thank you.
Any other questions?
Yeah. From my side, a therapeutic question. So you showed that the pathway goes via BIM. And so are there any therapies kind of, already out in the market that could be used in type 1 diabetes, or is it possible to target at that level?
To target BIM.
Yeah, for example.
It could be really complicated because BIM is involved in it's like the one of the last steps of different pathways that leads to cell death in general. And the problem is that the cells sometimes need to die, especially for infections or in fact, PTPN2 is also a target for cancer therapy. Because also, so for example, the higher levels of PTPN2 inhibit, for example, the drugs that target these cells that you want them to die. So going to these proteins like PIM or something like that, maybe for beta cells you can be saving beta cells, but you can add in other problems in the other cells. I mean.
Great, thanks.
Maybe to also add to that, so you for clinical application, you said okay, so maybe patients can with a certain risk gene, right? They carry a certain risk gene, you can treat, but so how do you imagine this to apply this in the clinic? Because then people need to be screened, right? Do you have an idea about this?
Yeah, my idea is that if in the future this kind of screening for polymorphisms is more common, that could be really interesting for the patients, because for example, PTPN2 polymorphisms are not only associated with type 1 diabetes, but also for rheumatoid arthritis. And this TNF-α treatment, both targets these two diseases. So, for example, if in this early screening of a patient, they discover that they have this kind of polymorphism, for example, they can start already with this anti-TNF-α treatment, and then they can have a better outcome of type 1 diabetes, but also rheumatoid arthritis. Or at least the medical doctor can have a better follow-up of the patients to be ready in case some of the symptoms start showing up, because you know that the problem of type 1 diabetes is when it's detected, we cannot do much.
Okay. And what is the proportion of patients with type 1 diabetes who actually have this polymorphism?
It's something that I didn't find, and I check in the bibliography and ask colleagues, but it's not described. Yeah, I think in some papers, but the problem is that there are several polymorphisms. I remember right now one paper that but it was a specific polymorphism. It was like a 5% of the patients that they were checking, but the number of individuals was not too big, so I'm not sure. I would like to know it. In fact, that would be interesting. And also, sorry,
no, go ahead.
Because this is something that in our lab we discuss it a lot of times. We also would like to know, especially people that they receive this TNF-α treatment. We like to know if they have any of these polymorphisms in the future. That could be really interesting.
And maybe we are at the clinical track already. So maybe I was wondering if, in your opinion, also testing other cytokines like the IL-1 beta or the interferon gamma will be interesting to test as well, because now you see already differences, right, between the two cytokines you tested. Do you think that others work the same, or are there any plans to test other cytokines?
In the context of PTPN2, I test other cytokines like interferon gamma or interleukin-1 beta. Interferon gamma, yes, it has a deleterious effect, which is already described by our team. In the case of interleukin-1 beta, it's not there is no effect. And in a more general context, without thinking only in PTPN2, we are already studying the individual effect of also the effect of individual cytokines in beta cells, and that will be published, I hope, soon. So we're working in that. We are really interested in studying the single cytokines, mostly because if you follow these kind of studies in beta cell models, most of the times when researchers want to induce beta cell death, they use a cocktail of different cytokines. And this could be a problematic because they are using cocktails of cytokines that they do different functions. They want, even if there are only one to test if for more beta cell death. It could be problematic using the cytokines that they affect different pathways. Yeah.
Great.
Sandra, but your question was blocking cytokines in the clinic or in vitro?
Well, of course, I guess the end goal is the clinic, right? But you first need to test in vitro to see if it's interesting or not.
No, I just come back from the immunology of Diabetes Society meeting. So blocking of interleukin-1 has been tried in the past and it doesn't work. So the big novelty is a study where they used an Australian group, used the baricitinib, which is a JAK1 and 2 inhibitor. So it blocks interferon gamma and interferon alpha. And over 1 year, they saw very good preservation of C-peptide, as good as anti-CD3. So I think the trend is to focus on interferons.
Okay, thank you for this addition.
That's interesting. And just a question, so the interferons, they're then also secreted by the immune cells within kind of the islets of Langerhans, or?
Arturo, go.
No, interferons are secreted by the immune cells that are infiltrated in the islets.
And what immune cells? So is it mainly macrophages also in type 1 diabetes, or B cells, T cells?
I guess there are T cells, but in that thing you are catching. I'm not sure. Not an immunologist.
I can help you. So the interferon gamma is T cells, interferon alpha is dendritic cells and macrophage. And beta cells produce very little.
Okay, thank you. See, Pierre has a question.
Yeah, thank you very much, Arturo, for this presentation of paper. And following up on this question, you said at the beginning you had a vicious circle between the beta cells and the immune cells, which are kind of triggering this increase of cytokine production. Do you think if when you perturb or delete the PTPN2 gene, you might affect also this crosstalk between beta cells and immune cells into that direction and maybe change the cycle you explained at the beginning?
If I delete the PTPN2 in beta cells, this cycle is going to be increased. It's going to be, so the cells are going to die way faster.
Okay. And do you think you have different crosstalks, like some cytokines or other products which are released by islet cell that could change the response of the immune cells around them, or is it just the classical death of a cell that will trigger the immune response?
No, another thing that we observe is that when we silence PTPN2, there is a more expression, for example, of different chemokines like CXCL10 that they attract immune cells. So that will be improve this vicious cycle. So for one side, what I presented, more cellular death, and then for the other side, there is more production of CXCL10. This is via the activation of the JAK-STAT pathway, and this will lead to more immune cell infiltration, and this cycle goes and goes. But of course, this is theoretical, because what I'm working in this paper is only in cells, in cell lines.
Okay, and by curiosity, can you do co-culture between immune cells and beta cells to show this? We are
doing that. we are starting to set the experimental settings for the co-culture. So yeah, it's an idea for the future.
Great.
Are there any other questions from the audience? If not, we might switch to a bit more career-wise related questions, because I was wondering, you made the switch between from doing a research more type 2 diabetes related to type 1 diabetes research. Was there a special reason for doing this?
Yeah, in fact, and it's not, I did not switch from type 2 diabetes, even worse. I mean, I work in obesity, so I became like an obesity expert. But the reason to switch is that, you know, obesity is a condition that involves a lot of different diseases, and it's a kind of complicated topic to study. And me, I wanted to focus more into a single disease. Because I thought that this I could add more of myself if I have a really clear objective. And I was always interested in the study of or what is happening with beta cells in type 1 diabetes. This was the only reason. And in fact, I think it was a risky jump, because now this is my second postdoc, but I consider it like my first postdoc now, because I changed completely the topic. But I think it was for good. Now I feel more focused in this, and I'm really excited with every time that we find something new. It's really exciting. So it was a good call.
So the research you showed today, does it contribute to your future career in the type 1 diabetes research field?
I think so.
So you want to keep doing this type of research for now?
Yeah, and in fact, it's what I said at the beginning, that this is just the beginning of a project that is continuing, and we are finding really interesting things that I would like to show in the future. But yes, I would like to continue with this in the future for sure.
Okay, and I was wondering how you experienced working in the 3 different countries, right? Because there probably are quite some differences between these countries. And what did you learn from this? And is this something you would recommend to others?
Starting by the last thing, I wouldn't recommend it. I will make it mandatory. Serious, because I think it's the best experience that anybody can have. Because the one thing is visiting a country for tourism, and another thing is living and working in a different country for everything. I mean, the language, the way of working is different. To be surrounded by international environment that is enriching you. I mean, in each laboratory that I learned something new. So yeah, I think it's an amazing experience that everybody, at least everybody that is starting in science, should have in mind.
Cool, thank you. Claudia, do you have any more questions, maybe?
Yeah, maybe also career related. So what I always find challenging is kind of in this stage where you are at now, kind of choosing the next step, because it's quite, it can be quite different from kind of what you choose. So how is your, what are your plans for the future? Where do you see yourself, or which career path do you envision for yourself?
So my idea, of course, this is theoretical. It's really hard to get that. My idea is to be a PI, to be a principal investigator, to have my own projects, my own grants, and just to develop something by my own, having being able to hire nice investigators and also train them and mentoring them. That would be fantastic. So this is what this is my focus right now. As I said before, I switch topics, so it's like I had to do a double effort to go back there. But my idea is to have a strong basis in the topic that I'm working right now in candidate genes in type 1 diabetes and publish papers and then getting grants for stabilization. That's my idea. But of course, this is a really complicated path, but we have to try. And
do you have a preference for a country to do this?
No, I just want to work in what I'm working. I don't care the country. Even if I had to cross the ocean, I cross the ocean is fine.
Okay, thank you. Anything more, Claudia, or that was a...
That's fine from my side.
Okay.
So thank you very much.
Just so one more call for the audience. Are there any questions for Arturo left about his research or about his career? I don't think so. So then I would like to thank you all for joining today's journal club. And I thank you, Arturo, of course, for presenting, and hope to see you all next time.
Thank you.